This part of the lecture is on the organelles that function inside the cell. So we just saw that in a film called Bioflix, and it went past quite quickly, so I think I'll re-show it again on Monday so that you can see it again. But let's just go over the functions of the various organelles inside the cell. So the cell is a eukaryote. The human cells are all eukaryote cells. That means they have a nucleus. Bacteria do not have a nucleus. So the nucleus is quite large. Some cells are anuclear. That means they don't have a nucleus. The red blood cell is an example of that. Some cells are multinucleate. They have more than one nucleus. So skeletal muscle is an example of that. Skeletal muscles are quite long, so they require more nuclei to service the whole cell with proteins that are required. The nuclear envelope is a 2-unit membrane. It's held together at nuclear pores, and the pores allow for the in and out of messenger RNA. So that exits through the pores. And nucleoplasm, that is the liquid that's inside the nucleus, has a chromatin, that's DNA wrapped around a protein. So DNA is only called chromosomes when they are wrapped for splitting of the cells. They're very condensed when they're chromosomes. Otherwise, they're not. They're just loose in the cell, in the nucleus. The nucleoli is a dark mass where ribosomes are produced. So there are many ribosomes. They consist of RNA. There's a ribosomal RNA. There's also transfer RNA, and those are produced in the nucleolus. Here's a micrograph of the nucleus. The nucleolus is quite a dark mass, and the nuclear pores are seen here. So this is formed by fractionation of a cell. So basically the cell is frozen and fractured, so it releases the indentation of the pores. The eukaryote cell is very interesting. It's different than bacteria in that the nucleus is surrounded by a double plasma membrane, but also there's lots of membrane-bound organelles inside the cell. So they function to separate what's inside them from the rest of the cell, which is good because the cell contains enzymes that tend to break down things. So it's good to have new proteins and DNA isolated from these enzymes that could conceivably break them down. So the rough endoplasmic reticulum is parallel flat membranous sacs covered in ribosomes. They are continuous with the nuclear envelope and the smooth endoplasmic reticulum. Thus, the whole thing is known as the endomembrane system. The endomembrane system. The rough endoplasmic reticulum is functional in the synthesis of proteins and phospholipids as well, and proteins of the plasma membrane. So some proteins are packaged and released from the cell. Some just stay in the plasma membrane. And some are even retained inside the cell. The smooth endoplasmic reticulum really does look smooth. It does not have ribosomes attached to it. It has cisternae. Cisternae are the shape of the endoplasmic reticulum in the cell. It looks like membranous sacs like that. They tend to be more tubular and branching, so they tend to branch off a lot more. Whereas the rough endoplasmic reticulum, if you were to look at that, the rough ER, it's also kind of stacked up like that, but it's studded with ribosomes. so it looks quite different. The smooth endoplasmic reticulum is responsible for the synthesis of membranes, of steroids, particularly in the ovaries and testes, and lipids. Also, the smooth endoplasmic reticulum is responsible for detoxification of toxins in the body. In particular, this, if you were to look at cells in the liver and the kidney, you would see that there's tons of smooth endoplasmic reticulum. It also stores calcium, particularly in skeletal and cardiac muscle, where calcium is required for muscle contraction. So this is what it looks like, the rough endoplasmic reticulum, which is studded with ribosomes, and the smooth endoplasmic reticulum. And they are also continuous with each other. It's all part of the endomembrane system. One sec. Sorry.